Good morning students. I am Ms.M.Aruna, HoD, Department of Zoology, JMJ College for Women, Tenali. Let us discuss about lymphoid organs. There are two types, namely primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs already we have learned. Now we will learn about secondary lymphoid organs. They are namely lymph nodes, spleen, Peyer's patches and tonsils. Secondary lymphoid organs, they are concerned with immune system. They contain lymphocytes. They are made functional. They are exposed to antigens. Poorly developed at birth and they grow progressively with age. Let us know about the objectives. To know the immune system, to know the structures of secondary lymphoid organs, to understand the functions of secondary lymphoid organs, to know the T and B cells, to understand the functions of T and B cells. Secondary lymphoid organs contains first one, a lymph node. Lymph nodes, they are complex, cellular, ovoid or spherical in shape. They are arranged on either side of the lymphatic duct or lymphatic vessel. In human being, they are bean-shaped structures. Its size about 1.25 mm in diameter. Its function is which filters the lymphatic fluid and also detects the antigen which are present in the lymphatic circulation. Then they are present all over the body. More number are present at joints, then mostly at groin and axilla. That means the leg and arms are attached to the body regions, contains more number of lymph nodes. This lymph node is a encapsulated structure. It is a bean-shaped structure. This capsule penetrates into the inner tissue to form a septa or trabecula. Here outer region contains many afferent lymphatic vessels which carries lymphatic fluid into this lymph node. Then one side we can see a concavity structure or indentate that is known as hilus, through which blood vessels goes in and comes out. That is the artery enters in, vein comes out. Then on the surface of this region we can see afferent lymphatic vessels. Many number are there which carries lymphatic fluid into this lymph node. Then this side a single efferent lymphatic vessel is there which goes out from this lymph node. This structure is divisible into three regions, namely outer cortex, middle paracortex, inner medulla. This outer cortex contains B cells. These B cells are gathered into bundles or groups. They get together to form a group. That group is known as primary follicle. Inside that primary follicle another rounded structure is there. Now it is known as secondary lymphoid follicle. Inside that another small rounded center is there, that is the germinal center. This is the site for multiplication of lymphoid cells. Then middle part is the paracortex, inner region is the medulla. Medulla has here lymphocytes are arranged along the connective tissue fibers. They are known as medullary cords. These medullary cords are separated from one another by a large space, that is the medullary sinus. This medullary sinus contains macrophages, plasma cells and phagocytes. Here this is the outer cortex, middle paracortex, then medulla. Cortex contains B lymphocytes. In addition to these, dendritic macrophages also there. Then in the middle part, paracortex contains T lymphocytes. Along with T lymphocytes, it contains antigen presenting cells. Then here MHC class 2 antigens are placed on the surface of the antigen presenting cells. Then inner part is the medulla. It contains B lymphocytes as well as T lymphocytes. Now let us know about the functions of a lymph node. Here it contains phagocytes which performs phagocytosis, which kills disease causing germ. As it has B lymphocytes, T lymphocytes, they perform humoral immunity as well as cell mediated immunity. Second organ is the spleen. It is a solid encapsulated organ which is placed in the upper abdominal cavity just below the stomach, close to the diaphragm. Here also it has a hilus is there through which artery goes in, vein comes out. Now we can see the transverse section of spleen. Here this organ is encapsulated. This capsule penetrates into the inner tissue to form a septa or trabeculum. Here this organ is divisible into two regions, outer red pulp, inner white pulp. Outer red pulp contains vascular sinusoids. These are the blood-filled sinuses. So here they contain plasma cells as well as phagocytes. So this spleen filters blood to detect the antigen which are present in the blood, whereas a lymph node filters the lymphatic fluid and it detects the antigens which are present in the lymph. And then it also acts as a graveyard. That means it destructs the old and worn out RBC cells and also performs hemopoiesis. That means it produces RBC cells during embryonic stages till 5th month. After that it will be taken up by bone marrow. So in the adult condition, during emergency, sudden decrease of blood time, it also takes over function along with bone marrow for the production of RBC cells. Then here arrangement of cells, lymphocytes, first described by a scientist named Malfigi. So this inner part, outer region is the red pulp, inner region is the white pulp. White pulp has lymphocytes, arrangement of lymphocytes in the inner region, white pulp. It is also known as Malpighian follicle, because this arrangement of lymphocytes first described by scientist named Malfigi. This white pulp gets separated from red pulp by a region that is the marginal zone. The inner white pulp contains B lymphocytes as well as T lymphocytes. B lymphocytes are gathered to form a bundle that is known as primary follicle. Inside that germinal center is there, which is for multiplication of lymphocytes. In between these follicles, many T lymphocytes are dispersed. Let us know, so this white pulp is arranged around the central arteriole that is known as periarteriolar lymphoid sheath, that is the pulse. Let us know about functions. It acts as graveyard, it acts as a reserve tank, also filters blood to detect the antigen, and as it has T lymphocytes and B lymphocytes to perform humoral as well as cell mediated immunity. Next we will learn about MALT. MALT means Mucosa-Associated Lymphoid Tissue. Here this MALT, a group of patches are dispersed all along the mucus layers which are present in the respiratory tract, alimentary tract and urinary tract. They are namely Peyer's patches, tonsils and appendix. Then another one, GALT. GALT means gut-associated lymphoid tissue. See, this is the Peyer's patch. Peyer's patches are formed because of gathering of nodules. They get packed below the mucous membrane to form an oblong elevated structure. Then here it is seen at intestinal region. Intestinal region has many microvilli are there at the surface. Here small cilia also there. But at Peyer's patch there are no microvilli, but it is enveloped by a specialized epithelium which is made up of M cells. M cells contains particulate cells which transfers the microorganism. This is causing germ from lumen of the alimentary canal into this dome shape or oblong shaped structure. So when this antigen enters into this dome, to stimulate that inner part to produce B lymphocytes as well as T lymphocytes. Here these are the B lymphocytes, these are the T lymphocytes. So after entering this pathogen into this region, these B cells enters into efferent lymphatics and the B-centric lymph nodes. After that reaches the thoracic duct and enter into the blood. After that immediately it re-enters into the lamina propria. Here they get matured into IgA plasma cells. These IgA plasma cells secrete secretory IgA molecules. Now these IgA molecules enters into the lumen of the intestine to neutralize these microorganisms like virus, bacteria or any disease causing germ, and also blocks that microorganism by making a paint, antiseptic paint around that microorganism. Now we'll discuss about tonsils. Tonsils are the mucosa-associated lymphoid tissue. These tonsils are their resemblance with thymus, because these tonsils at the time of birth they are small in size. Its size increases when the age advances, whereas all secondary lymphoid organs are poorly developed at the time of birth. After that they increases their size when the age advances. Here it is a reverse condition, it shows reverse. Here it is a lymphoid tissue encircling the pharynx. It is a follicular structure, contains lymphocytes, macrophages, mast cells. Germinal centers appear in response to antigen. It is protective role in URI, means upper respiratory tract infection. When air enters into the respiratory tract, immediately tonsils detects that microorganism and traps it, and after that it eliminates it from the respiratory tract. Tonsils are three types, namely palatine tonsils, lingual tonsils and pharyngeal tonsils. Palatine tonsils, they are present at the palatine region. Lingual tonsils, they are present at the base of the tongue. Pharyngeal tonsils, they are at the back of the pharynx. They are also known as adenoids. Now look at the picture of this tonsil. Here the gathering of B lymphocytes, they are the primary follicle. This bundle is known as primary follicle. Then inside, when germinal center is occupied, now it is known as secondary follicle. Here we can see mixed seromucous, mucous gland, tonsillar crypt, diffuse lymphoid tissue. Recapitulation. We have learned Objectives: secondary lymphoid organ structure, lymph node structure and its functions, spleen, its functions of spleen, Peyer's patch and tonsils. I hope you have understood well. Thank you one and all.